What kind of dog do you find in a bakery? Hot dog?
That's a bakery. No.
Bakery. A purebred. What did the baby pig call his mother's brother? The baby pig, what did he call his mother's brother?
Twinkle.
Oinkle. Nicely done. Nicely done. What do you call a cow with a twitch? What do you call a cow with a twitch? Beef jerky. Well done in the back. Okay. Y'all ready for Wednesday? No? Shouldn't be a surprise. We've been talking about it for a while. All right, so I'm gonna have office hours tomorrow on Zoom, 10:30 to noon. And I have office hours on Wednesday, either Zoom or in person in my office in Wetop from 11 to noon. Okay, you get your last minute questions answered. We have a review session tomorrow, 6 to 7 p.m. in Bio 256. Review session tomorrow, 6 to 7 p.m. Bio 256. We have exam 4 on Wednesday, and your bonus number 4 assignment. If you haven't already done it, it's due tonight by 11:59 p.m. Okay, I've actually had some folks emailing me saying that they're confused about when assignments are due because it's not like showing up on their BD Learner calendar. So, I mean, this is one of the reasons you all know if you come to class, I literally announce what's happening this week, right? And then, has everybody been getting the BD Learner announcements that push to your device? So you even got one yesterday reminding you of the quiz, right? Okay. So I'm doing everything I can to communicate to make sure you're not missing stuff. So if you have a friend who's either not coming to class or not watching the lectures online, let them know, you know, there's a lot of different modalities to get this information. Okay, bonus 4 has up to 20 points available to it. That's the biggest so far. You have an hour and a half to do it. So it's technically timed. So I think it would be wise to preview the paper on muscular dystrophy so that, you know, you highlight some salient points. On these papers, folks, a good rule of thumb is read the abstract first. Like the big paragraph right underneath the title. It gives you an overview of what's happening. Kind of, it's daunting. You pick it, you look at it on your device and you're scrolling forever and you're like, holy crap. Okay, and you read the abstract, it'll give you a synopsis of what the paper is about. Okay. Any questions on administrative things before we get started? So we just have a little bit of material to cover today, and then we'll use the remaining time for a review session. Just like what we did last time. Seemed to be positive. I can't go an hour and a half like we did last time, or whatever the timing ended up being. And I don't know when we get kicked out of this room. So we'll kind of try to end pretty close to class being over. But there's review sessions tomorrow night. I have office hours. Okay. I just got carried away last time and lost track of time. And before I knew it, we were like in a review session for an hour plus. But you guys kept asking questions. I wanted to back up slightly and just kind of reiterate these systems of energy use. And we'll do this kind of review style. So someone tell me this immediate demand. Tell me some characteristics of this immediate demand of energy as you've been studying. You got the cheat sheet up here, so you can literally read the slides. But you tell me, what are some characteristics about immediate energy demand? Where are you getting it from? Creatine phosphate, even more basically, that's true, that's a detail. Even more basic than that is phosphate donations. You're lending phosphate, or you're stripping phosphate from one molecule, bringing it over to another to make adenosine triple phosphate, adenosine triphosphate. Make sense? That's all that's happening here, is you're just shipping phosphate or stripping it from one molecule, putting it on another. This gentleman's correct. Creatine phosphate is one example of that. Myokinase would do the exact same thing. This is for energy demand that's fast. Quick sprint, less than 10 seconds. Next up, tell me what's happening on short-term demand of energy. Give me some characteristics. Let's talk about timing. How long does this last?
30 seconds.
10 to 30 seconds. Yeah, maybe up to 40 seconds max. Okay, am I gonna have up to 30 seconds, up to 40 seconds as an option? No, I won't do that to you. It'll be pretty obvious, right? It'll be like 10 seconds or less, probably something like 30 to 40 seconds or less than a minute, maybe something like that. Beyond a minute is what? Or after 40 seconds, really beyond a minute. What kind of energy is here? Like the answer is literally written on the slide, just in case you're curious. Aerobic. All right, maybe you're just shy today. We've been together all semester. You're gonna be shy now? Okay. Aerobic respiration. Somebody explain that to me. What does that mean? I'm confused, Dr. Keller. You're trying to be tricky. You're calling it aerobic respiration. I'm confused. What does that mean? Utilizing oxygen. Higher or lower amount of energy than anaerobic, or breaking down glycogen or glucose. Higher or lower aerobic? Everyone? Higher. Well done. Faster or slower energy access? Anaerobic versus aerobic. Anaerobic, that just means without oxygen. Which is this faster, the anaerobic, or slower than aerobic? Everyone? Faster. So faster, but less energy. Do you see the difference? These are like two different tools. Now, unlike your automobile, unless you have like a continuous variable transmission, you won't feel your body switching fuel sources. There's not like an end of one and the beginning of another. It kind of comes up and then tapers off. The other fuel source picks up, tapers off. Then the third fuel source. You guys understand the analogy? Like your transmission on your car, if you're like racing it off the line, you feel it kind of shift if you're driving automatic. So there isn't these discrete moments in time, but at the end, whatever you do at the end, there's this oxygen deficit. What is this oxygen deficit? Let's talk a little bit about this. What does that even mean? At the end of a 2-minute run, you don't stop running and then you're just like, you're running. You stop running, you're like, very good. You're breathing heavy for a little while. The longer you run, the longer the recovery or the repayment of that oxygen debt. What's happening there physiologically? Why are we even talking about this?
Like replenishing the oxygen that's to sustain that motion.
Okay, you're replenishing the oxygen you just used for that exercise activity. Yes. And this is foreshadowing the next semester. That's why I'm talking about it. There's this huge system in your body called the bicarbonate buffer system. You'll learn about it in the cardiovascular respiratory section. Okay, and the idea is the way that you eliminate excess acid, specifically when you're using the lactic acid glycogen system, the byproduct of breaking down glucose through glycolysis is you make lactic acid. As the name implies, it's acidic. That means it has a lot of free hydrogen in it. And so if you use this system first and you burn all that fuel and then you shift to another fuel source, when you're done with your activity, you actually have to eliminate that excess hydrogen ion. So a way that you do that, a very effective way you do that, is you take the free hydrogen ion and you bind it to bicarbonate in your blood system. It moves to your lungs and it turns into water and carbon dioxide. And you breathe it off. You breathe onto a mirror through your sunglasses. What do you see? See vapor, right? That's the water vapor. If you put a probe, you'd be measuring CO2 and you can see visibly the water on the mirror or your sunglasses. So this oxygen debt is you're blowing off the excess hydrogen ion until everything stabilizes. I just wanted to finish the story. Okay. Let's shift gears to smooth and cardiac muscle. We're going to go pretty quick through this. I'm going to just kind of compare and contrast based upon what we know with respect to skeletal muscle. So in general, why are muscles contracting? You tell me, why are they contracting? Hope you move. Perfect. How are they contracting? How are they contracting? How do they do it? Excitation, contraction, coupling. Maybe you could tell me a couple of components that are involved in contraction. Are there a couple of key proteins that you might be able to describe? Sliding filament model with the myosin and the actin, or the thick and the thin filament would probably be more accurate. Because the thick filament, this was a question that was brought up at the beginning of class, before class, the thick filament is exclusively myosin, right? And the thin filament is made up of what? Troponin, tropomyosin, and actin. Which of those four proteins are contractile proteins? Which of those four proteins are contractile proteins? I'm going to develop a crick in my neck looking over this way, so help me out. Actin and myosin are contractile proteins. Which of those proteins are regulatory proteins? Then have only two options left. Troponin and tropomyosin. Okay. If we look at smooth and cardiac, you're going to see a lot of the same motifs, but muscles in general are cell rich. They have a lot of cells in them. And they're extremely well vascularized. And that should make some sense because if we need a lot of energy to run a mitochondria for long-term contraction, based upon the energy topics we just talked about, you need oxidative phosphorylation or aerobic respiration to happen. If you need aerobic respiration to happen for muscles to continue to contract, you're going to need mitochondria on board. You're going to need oxygen to be supplied. How do you get the oxygen to the muscle? How do you get the oxygen to the muscle? What's that? Blood. You perfuse it with oxygenated blood. So they're well vascularized. They're elongated cells. We call them fibers. Okay, on the exam, I'm pretty certain every time I said muscle fiber, I put cell after it. But if I didn't, you shouldn't be confused by that nomenclature. We've talked about it a lot. And they have these modified cytoskeletal proteins for contraction, like actin and myosin. We have three types of muscle. This is going all the way back to unit 1. We have skeletal, smooth, and cardiac. Skeletal muscle, what we've been in. Voluntary control, attaches to bones by tendons. Just a quick review. What is the Golgi tendon organ? Where does it live? You're like, oh, wow, where did you come up with that? Well, I just saw the word tendon. That just made me think of it. The Golgi tendon organ, where does it live? The Golgi tendon organ, where does it live? Oh, it lives in the tendon. Well done. Nicely done. It lives in the tendon. What does it do? It monitors what? Muscle tension, great job. Does it make sense that you would have it sitting in the tendon to measure tension of the muscle? Because that's sort of the funnel point, if you will, right? Okay, now back to the lecture. Elongated, multinucleated, and striated. Now we know a lot of the background of some of these terminologies from Unit 1. In the beginning of the lecture, you just memorize this stuff, I guarantee you. You're like, striated. I just had to memorize striated. Now you actually understand why it has a pattern to it. Make sense? That's the difference between the heavy and the light regions, like the dark and the light regions on electron microscopy that we saw. Multinucleated, we've seen that. It has many mitochondria, high energy demand. Okay, cardiac muscle under involuntary control. You don't sit there going beep, 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 beep. He's passing out the exams. Beep, 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 right? It's just, it involuntarily just rises, right? That's just some of you told me, like, you give me hypertension, Keller. Like, it's not intentional, okay? Or some of it might be, because I'm actually trying to prepare you for the next phase of your career. And this is a lot of what that's going to look like. Examinations. You're going to be examined for the rest of your life. Okay, awesome. Branched, single nucleus, but they're also striated. So if they're striated, does that tell you that the actin and the myosin are arranged in a very similar fashion as skeletal muscle? Yeah. Make sense? So in the beginning of the semester, in lab and lecture, you just memorize these little things. That one's striated, that one's striated, and smooth muscle's not striated. Did anybody really know why in the beginning? Now we kind of put the whole picture together. So we'll talk about smooth not being striated. So yes, the actin and myosin are arranged very differently. These have intercalated discs. I've already tested you on a lot of this stuff. I'm just trying to put the whole story together. Now smooth. We find these on the walls of internal organs under involuntary control, right? You're not like peristaltic move, move the food down, move the food. Oh, too fast, right? It just doesn't. Uninucleated, they're in a fusiform shape. So now we start, you remember, oh yeah, they were fusiform. I remember some of the stuff from the beginning of the semester. I had no idea why they don't have visible striations. It's just my lab instructor told me skeletal and cardiac are striated, smooth is not. Fine, I'll just memorize it. I have no idea why. Sometimes you just have to learn things and then you fill in the blank. Okay. So let's dive a little bit into sort of some of the differences here. We look at cardiac muscle. There's no nerve stimulus required. So that's hugely different because we've been focusing on nerve stimulus, synapse, postsynaptic potential where it depolarizes, right? So let's just refresh. You get a nerve signal coming down with a depolarization event. You release acetylcholine at the synapse. It binds to its receptor on the other side, and it opens sodium channels. And which way does sodium run? Into or out of the cell? Into the cell, and it causes what? What kind of event? An action potential, also referred to as a depolarization, right? Okay. So here you don't need a stimulus because these are autorhythmic. You'll learn more about autorhythmicity next semester in 202 with cardiac. And so I'm just kind of priming the pumps for next semester. This almost exclusively relies on aerobic respiration for its energy source. Does that make sense to you? Yeah. So if you cut off blood supply to the heart, in other words, you cut off oxygen supply to the heart, can the heart continue to beat for very long? No. So that's the whole premise of a heart attack. We call it a myocardial infarction. Now, interestingly enough, the heart will run for a few minutes. It'll shift over to glycolysis. And one of the things you can measure in the bloodstream are glycolytic byproducts to determine if there was a heart attack in the patient. And you can actually you can measure if it's a serious myocardial infarction. You can actually measure troponin levels, cardiac troponin specifically in the bloodstream, because the cardiac troponin starts to degrade really fast. Fascinating, huh? It's not similar, it's similar to skeletal muscle troponin, but it has its own isoform, and you can measure that through a blood test. And many of you will do that. Like, hey, we're going to test this patient for cardiac troponin. You're going to go, I actually know why. Because we want to know if they shifted from aerobic respiration to glycolytic pathways, and if it was a major myocardial infarction. Okay, so I get a lot of times I get students asking, why do I have to learn this crap? Okay, well, hopefully you'll start to see as you put together, you have to have a baseline of information before you can start putting the story together. And then 202, I think you're going to find really fun. And as you move on in your studies, like this is the foundational class. This is non-mitotic, meaning they do not divide. And the central dogma is just terminology that says that's the well-accepted theory. Now let's look at smooth muscle. I'm just kind of starting from the bottom, working up. They undergo mitosis. They can actually divide and repair, whereas cardiac cannot. Skeletal muscle goes through mitosis. It'll divide and repair. You tear a skeletal muscle, it actually repairs really fast, faster than bone. Like a muscle tear is a great injury for an athlete because you don't want to tear a tendon or a ligament. It'd be much better to break the bone. You're like, what? Or tear the muscle because it'll heal very quickly because muscle and bone are well vascularized. Tendon and ligaments are not. And tendon and ligament tears take forever to heal, and many times they don't fully heal. This actually undergoes mitosis. It has a fusiform shape. That means it looks fat in the middle, thin on top, thin on the bottom, kind of like a stretched out football. That's fusiform. We don't have striations. We don't have Z discs. We don't have T tubules. Like, wow, this is super different. But if you can conceptualize that statement of we don't have striations, we don't have Z discs, we don't have T tubules, if you can conceptualize that, well done, you're a nerd. Because you're just like, oh my gosh, how does that even exist? It's blowing my mind how there's no T tubules or Z discs. And those are my dinner conversations at the table. And all my family's like, Dad, can we talk about something else? Okay. So if you're conceptualizing that, this stuff's starting to sink in, because that actually is really kind of cool. So where does the calcium come from then? Calcium is different. It comes from the interstitial fluid. So instead of storing it in a sarcoplasmic reticulum, you bring it from outside the cell. But it still has actin and myosin, and you can see that's these two blanks. You can see this drawing over here where we've got a relaxed form. Here's our myosin, right? And then here's our actin. And so it's more of like a net. It looks more like a fishing net. And then when it contracts up into like a ball. But because it looks more of a network pattern, it's not organized like skeletal or cardiac, so you don't get the striated pattern. You with me? But you still have cross-ridge formation and overlap. Your calcium just exists outside of the cell. It doesn't store it inside of the smooth muscle cell. We've got multi-unit, which is on the left, and we have, multi-unit on the left, and we have single unit on the right. So what this is referring to is the multi-unit, where you can see that each individual spindle cell has its own nerve that's actually going to it. Whereas over on the single unit, like visceral muscle, you actually have one nerve that comes down and it serves this entire population of smooth muscle cells. So each one of these sort of long spindle-looking structures is a smooth muscle cell or a smooth muscle fiber. And then really our last difference is in the way the autonomic nerve fibers exist. We have what we call these varicosities. So you kind of see this autonomic nerve fiber coming in, there's these little bulges, right? They look like little pea pods or something. And if you zoom in inside of them, you'll see mitochondria, and you'll see synaptic vesicles. And so this is actually where you get this innervation and the excitation contraction coupling that we learned about in skeletal muscle. We refer to these more as varicosities and diffuse junctions, just like what we saw diffuse junctions at skeletal muscle. But the varicosities are sort of a little unique package like the synaptic knob. Kind of cool, huh? So as you study and you move on, if you move into research, you look at other organisms, you'll see very different motifs of the way their nervous system interacts with their locomotion. A lot of different motifs in architecture. And we just are focused on 3. 2 are pretty much the same, skeletal and cardiac. And then smooth is the one that's really unique. Questions? All right. I've got a couple of more pieces of information. This is the last slide here, and then we're gonna, on this little unit, we've got a couple of questions. I'm gonna finish out with fiber typing. Smooth muscles, how do they contract? Well, we go from a relaxed state to a contracted state, and we've got calcium gates that are on the plasma membrane. So they're bringing calcium from outside the cell versus from the sarcoplasmic reticulum. These are voltage regulated, or they could be ligand regulated or mechanically regulated. So ligand regulated could be a neurotransmitter. Voltage gated could be a change in action potential. Mechanically gated could be if something presses up against them. Okay, so 3 different ways that calcium can actually move into the cell. Calmodulin. So calmodulin is a variation where you get the binding instead of troponin on calmodulin. So this is the troponin substitute for smooth muscle. And then from a characteristic, these have a long relapse period. That's what latent means. They're very slow to contract, and they're also very slow to relax. So if you look at the action potential, if you remember the action potential of a nerve, now that action potential firing is very characteristic of how skeletal muscles contract. They follow the same motif, very fast, and then they relax. With smooth muscle, it's more of a sinusoidal wave of contraction and relaxation. And you're thinking about like peristalsis, where you swallow a food bolus, or you move food through your alimentary canal or your GI tract. In peristalsis is you contract behind the bolus, and you kind of do this, and you move it this way, but it's nice and slow. Think about it. You don't want to move food through your digestive tract too quickly, because you want to absorb as much of the nutrition and calories as possible, right? So a couple of strategies for patients if they need to move things quickly is you stimulate GI motility, and usually that turns into diarrhea for the patient, okay? If you need to slow things down, right, you'll slow contraction. And if it goes too aggressively, now the patient is constipated because too much of the fluid is absorbed out of the chyme, which is what you call food once it leaves the mouth and it's, into the digestive tract. It's called chyme. Question?
Is the terminology for different parts of muscle the same for smooth muscles, or because you call the plasma membrane and not the
Sarcolemma. Yeah, so in smooth muscle, it's still interchangeable. Still interchangeable. We would be comfortable to say the sarcolemma of the smooth muscle or the plasma membrane. Yeah. But some of the terms that are not interchangeable, like calmodulin is the substitute for troponin. They don't have troponin. They just have a different molecule. Okay. 1997. It's a great year. Many of you weren't born, but it was a good year, I promise. This Denmark runner set the record for the 800-meter dash, 1 minute, 42, 0.67 seconds. As he was finishing the race, which energy source predominated in fueling his leg muscles? I just want you to think about it. I just want you to think about it. Take a volunteer. You need to tell me why. Okay, volunteers. I saw a hand right here first. You gonna pass? Okay. The back or middle section? Aerobically generated ATP, it would be. Why? Y'all hear that? C, aerobic, because it's greater than 10 seconds, which eliminates A. And it's greater than 40 seconds, which eliminates B. And D also would go along with A, right? Because that's a quick phosphate donation. Because your timing is well over a minute. Okay, well done. How about this one? This picture, you see the arrow? So it's going like it's a bicep curl, essentially. It's an example of an isotonic and concentric, isotonic and eccentric, isometric, or, well, you're not allowed to say D on Wednesday, okay? Think about it. Volunteer. Isotonic and concentric. Why? Can you explain? Muscles are shortening. That's the concentric part. Agreed. And it's changing the length of the muscle, right? Because it's shortening. Okay. How about this one? This was my water bottle example. All right. Water bottle. Okay. Think about it. Give people some time. Just give people some time. If we go too quick, sometimes people are trying to figure out the answer, which I want them to do. What is she doing here?
The isometric, right? Because there's no like change in the length of the muscle.
It'd be isometric because there's no actual change in the length of the muscle. Say, I mean, ideally, right? Ideally, she's not coming down the hallway, curling your food. Okay. How about this one? So really look at the middle picture to the bottom picture, and then he goes back up to the middle picture. The top picture is just getting set up. Middle to bottom picture, back up to middle. I need a volunteer. B, why? No, you're right, it's B. It's B. Why is it eccentric? Is the muscle shortening or lengthening? It's lengthening. Okay, so isotonic and eccentric. Okay. Well done. Did that make sense?
Just for clarification, are you able to say that concentric would be like a flexing and eccentric would be an extension?
Not always, because flexing and extending is usually about a joint. So with concentric, usually it's best to say the muscle is shortening, it's concentric. If the muscle is elongating, it's eccentric. Okay. So for example, if you were trying to control your descent coming down a hike, like down Humphreys Trail. Anybody hike Humphreys Trail? Okay, you're coming down Humphreys Trail and you're trying to control your descent. Think about what's happening at at your quadriceps on front, as well as your hamstrings in the back. Okay, so as you're controlling your descent, right, you're actually putting the brakes on, so you just don't face plant. Right, and you're firing your knee flexors, right? Because you're trying to absorb the shock as you come down. So think about terminologies as it relates to the action on the muscle. Eccentric versus concentric. All right, different fiber types at the very end here, and then we're going to move into review. We're almost done. But a lot of this, especially for those of you that are kind of exercise science, exercise physiology, physical therapy, and I actually think this is great even for our nursing, dental hygiene, as well as, you know, pre-med and researchers. You're understanding a lot about the way we categorize muscle, how it works, the different fuel systems that the muscle uses to generate the ATP. So with slow muscle fibers, and we're just going to really talk about two types, slow and fast. And you'll learn that there's an intermediate type as you move on in your studies. But for purposes of our class, slow and fast. Slow is usually considered red muscle. And the reason we call it red muscle in the literature, it's kind of an old terminology, but it has a lot of capillaries and a lot of mitochondria because they're slow oxidative and they rely on aerobic respiration to generate their energy source. So there's a lot of mitochondria, there's a lot of blood flow. Histologically, they look darker in color, reddish in color, like a dark red. And you're seeing histologically through a muscle belly, fast glycolytic, which is more of the color that you're used to seeing, and then this really dark red stain. And that's the slow oxidative, the SOs. And they're slow oxidative because, or they're dark in their color because of the richness of the blood supply and the mitochondria to generate the ATP necessary. But if they run off of aerobic, they have less power than the muscle we're going to look at. They're usually smaller fibers, and they have this high density of mitochondria, myoglobin, and capillaries. And I ask you why. We kind of already talked about it, but that's the energy source, right? Our postural muscles are slow oxidative. You don't want them fatiguing. Postural muscles. You don't want them wearing out. They need to last all day long. We also refer to these in the literature as type 1 fibers. So they kind of have three different names. Red muscle, slow oxidative, type 1 fibers. But all of those terminologies should make sense if you understand why we call them that. You with me? Let's contrast that to fast muscle fibers. They're usually larger, therefore quick bursts of energy. Their names are referred to as white muscle versus red muscle, just because of their staining color. It's more pink. I don't know why we call them white. It's more of a pink, but whatever. Fast glycolytic fibers, type 2 fibers, high power, but they're quickly fatigable. They utilize the creatine phosphate and the glycogen lactic acid pathways that we talked about. So this should all kind of make sense. Now, in your body, all muscles have some blend. It would be silly for them not to, right? It'd be silly for them not to. You have slow oxidative for posture muscles. If someone's like, hey, Rob, catch this backpack. Someone throws me a backpack and we just fall over, right? Because I can't withstand, I completely just fall over. So there are still fast glycolytic and postural muscles. And this chart, it's an old chart, but it still points out the take-home message, where you're going to have all muscles having a blend, but you're going to have certain training specificity based upon different types of activities, which will make sense. So if you're a sprinter and you train as a sprinter and your training specificity pushes you a sprinting pathway, you're going to see that your fast glycolytic, it's more of a 60-40 blend. And this is very male centric. I apologize for that. This is just the textbook that they had at the time. It's an old chart. I need to update this. So there's no female example here, which I apologize for, but with males, you can see that it's a little bit more 50-50, maybe 45-55. You push it 60-40 for sprinters. Swimmers, marathon runners, you see the oxidative fiber type is actually predominating. And we always get the question, well, is that genetics or is that training? And the answer is yes. It's both. There definitely are individuals that are born and they have a predisposition to certain types of muscle fibers, and maybe they're a little better at those activities. They train in those activities, they start winning in those activities. So that positive reinforcement pushes them into long distance running. And just the opposite, like I get bored after like 2 minutes of running. I have attention deficit disorder. Okay, well, maybe you should be a sprinter. It's over quick. Okay. Adapting to exercise. I mean, this handsome devil, look at this guy. Wow, what are you giggling about? How big his muscles are? Sorry, that was inappropriate. Muscles hypertrophy. Okay, so that word means that they enlarge. Muscles are enlarged. The picture's goofy, but you'll never forget it. And red muscle adds more mitochondria and more myoglobin as it enlarges. But you have to be careful, especially, not saying this, but especially with athletes that utilize muscle building regimes like steroids. You'll hypertrophy skeletal muscle, but you'll also hypertrophy cardiac muscle. They're very, very similar muscle types. And that's why with our athletes, where you see significant hypertrophy of the muscle in a very short amount of time, you worry about the cardiomyopathies that take place. And if you doubt what I'm talking about, just Google Arnold Schwarzenegger's history of bodybuilding and his surgical history. He's actually had a couple of heart surgeries that are very under publicized. All right, last couple of slides. Fatigability. So this is different than psychological fatigue, right? So many have asked these questions a lot. Like there's a lot of athletes in the room, a lot of people that perform, a lot of people that have a craft that, you know, maybe you don't compete, but you're pretty into your craft. Maybe you're a rock climber and you don't compete, you just do it for yourself. You're a mountain biker, a cyclist, you don't compete, you just do it for yourself. You know, there's psychological fatigue. This is different. We're talking about physiologic fatigue. Physiologic fatigue, where the muscles just wear out, and that was a question that was posed actually earlier in this lecture series. There's a lot of different options. Okay, so number one, you could have inappropriate calcium balance, where you're losing calcium. You could run out of glucose or glycogen. You could have an ATP shortage. You could have potassium that's being released that lowers the ability of excitation, right? You hyperpolarize, so you can't excite the membrane as easily. You run out of recycled acetylcholine. So here's a question for you. What was the enzyme that breaks down acetylcholine? Acetylcholine esterase. Remember that? It's basically ACH with a big E after it. So that's an important enzyme to break down acetylcholine. If that activity is so high and you can't bring in more acetylcholine quick enough, you could have fatigue of the muscles. I get this question a lot in other courses, like what about lactic acid overload in the local muscle? Remember that bicarb buffer system I told you about of exhaling, basically hyperventilating? It is extremely efficient, so that's typically not a limiting factor of that lactic acid burn. It burns for a while, but you'll start huffing and puffing pretty quickly. You'll blow off that excess hydrogen very fast, and you'll buffer that. So that's usually not a rate limiting step. So We talked a little bit about this oxygen debt, and we're going to end on this. And then I think I have one quick question for you, and then we're into review. After I run, after I bike, after I swim, whatever it is that you do, I'm still breathing heavy for a good 30 seconds to a minute to 2 minutes. And this is that oxygen deficit that we talked about, where you've got excess lactic acid that ultimately makes it into pyruvic acid, but this takes time. It needs to be converted back into glucose in the liver. And so, as appropriately was mentioned earlier, you have to replace your oxygen reserves that you've depleted, and you actually need to replenish your phosphates that you might have stripped off of other molecules. You need to move the oxygen around using transfers of different proteins like hemoglobin in the bloodstream, myoglobin in the muscle. So hemoglobin is the molecule that carries oxygen in the blood, and myoglobin is its counterpart in the muscle, which has a higher affinity for oxygen than hemoglobin. So it attracts it right off the hemoglobin molecule. So all of that has to be replenished. So you breathe heavy in this oxygen debt to replenish what you've lost. Make sense? Once it calms down, you're breathing normally. The more you train, the shorter your deficit period. Right? And you've seen this in your patients. Some of you are CNAs taking care of patients. They get up, they walk to the bathroom. They can still go to the restroom on their own, which is great, but they're breathing heavy. And you're like, wow. Or they can walk upstairs, but it takes a while and they're huffing and puffing when they come up and come down. Okay. So that's what's happening here is your body is working hard to make sure that that deficit is replenished. Okay. Last question. When a muscle's at rest, which of the following is not associated chemically with the others? And I read this earlier and I was like, oh, it's kind of worded a little interesting, but this is perfect because some of you are telling me that my questions are worded uniquely. Hang on one second. Let's wait for it and then I'll have you answer that. So we're at rest. Which of the following is not associated chemically with the others? Myosin, B. Not associated with what? You hear that? Myosin, B. Because at rest, myosin's the thick filament. It's not associated with the thin filament. The other three are on the thin filament. Make sense? I can see how some of you might say, well, isn't it two of those? Isn't actin also not associated with myosin? I think you could probably make that argument of, hey, well, if myosin binds to actin, doesn't actin kind of have to bind back to myosin at some level? Sure. Food for thought. So I would take E or I would take B. Make sense? Questions over the lecture? That's all the new material. You have a study guide. It's been up for a week plus. We had a review session tomorrow evening. We had office hours tomorrow, Wednesday. We have about half an hour remaining in this classroom. What would you like to talk about? You guys are good? Should we start the test now or? Whoa, that got a response. Yes, sir. The whole contraction process. Yeah. Oh, no, that's fine. Let's go ahead.
Like H-zone and D-line, those are, I just don't know what they are.
Okay. Okay, let's do this. Let's pick a couple of topics and get a show of hands. So you want to go over a picture of the sarcomere and the different zones. You want to go over sliding filament model and how things move. You want to go through excitation, contraction, coupling. Those are 3 separate topics. So let's do topic number 1. You want to look at the picture that has the labels and see where everything's at. Raise your hand. Okay. You want to go through excitation, contraction, coupling. Okay. Sliding filament model. Okay. So it sounds like excitation, contraction, coupling has it. Let's start there, and then we'll work our way backwards. How's that sound? Is that fair? Okay. Start here, excitation, contraction, coupling, sliding filament model, and then we'll do the diagram labeling. How's that? Does that work? Okay. All right, excitation, contraction, coupling. So this is a concept where you're basically saying, if you want to move up, you want to reposition, you do it. You know, some folks have left, so there's more space. You want to move into a different spot. So this is just a stepwise walkthrough of how we've got the signal coming from the nerve. Okay, that was last unit. Signal comes down from the nerve and reaches the end of the nerve in a space known as the synaptic knob. A structure, not a space, a structure. And that's this. I feel like it kind of looks like an alien hand. Okay, because it's not reaching out like this. It actually has a base to it. But this is the synaptic knob, where this is the nerve coming down, and this action potential, you see it's myelinated, so it's going to be going quickly. And in the different bare areas, the nodes of Ranvier, you have the movement of ions. You have sodium moving in, and then you have potassium moving out. And that was last unit. This myelin sheath also covers the end knob and envelops it over the gap or the synapse, so that when you release neurotransmitter into the synapse, it doesn't dissipate out too quickly. It really has a maximum opportunity to bind to its receptor on the postsynaptic side, otherwise known as the motor end plate. Because this on the bottom is a muscle. And so we refer to it as a motor end plate because it's a motor pathway to do something, like movement, a motor efferent output. If you blow this up in higher magnification view, you can see the acetylcholine receptor in purple. It's got a channel down the middle. It has 2 green acetylcholines bound on either side. Everybody on board there? So then we're going to go from here. We're going to go to this picture where we've got acetylcholine binding, opening the channel, allowing for sodium to rush in, and then ultimately potassium to leave. Sodium rushing in creates an action potential on this side, which is the muscle membrane. The muscle membrane is a plasma membrane. It has another name. It's called the sarcolemma. Question?
Separate and sodium channel and potassium. Why are those in the
So it's a great question. So here, we're talking about an acetylcholine channel that allows sodium and potassium to move. That's just at the local area. And the question is, well, if this is already moving ions, why do we need these separate voltage gated channels that you show later? I'm going to show them in a second. And the reason is you need to propagate that signal quickly down the membrane. And you're not going to have these acetylcholine channels anywhere other than right here in the junctional folds. So beyond the junctional folds, it's somebody else's job. And the other person's job are those voltage gated sodium and potassium channels that we learned about in nerve physiology. Answer your question? Okay. So that's here. So at the level of the synapse, you've got the acetylcholine that can initiate the sodium signal. And then you need a voltage difference over here to stimulate these voltage regulated ion channels that we learned about ad nauseum last unit. And that's what sends it down the membrane. I think the next picture, yeah, shows this arrow coming here. That arrow is being propagated by ions moving at this membrane right here. And those ions that are moving are the sodium ions rushing in and the potassium ions leaving more slowly to repolarize. Okay. So now we're here. We come along the membrane and we dive down a T-tubule. What is the T-tubule? Is the T-tubule a structure, a protein, a molecule? What is it? It's a structure. The T-tubule is transverse tubule. It's an extension of the plasma membrane or sarcolemma that dives deep into the muscle itself, so that that action potential can make it in to the middle of the cell where you have myofibrils, actin and myosin. When that comes down here, conveniently, there's structures on the other side of the membrane that are inside of the cell that are part of the sarcoplasmic reticulum, and these specific structures that parallel the T-tubule are referred to as the terminal cisternae. So same question, is the terminal cisternae a protein, a molecule, or a structure? It's a structure, and it's a structure that's connected to or part of what? The sarcoplasmic reticulum, which is basically a modified what? Endoplasmic reticulum, which is just a structure from a normal cell that you learned about in basic biology.
So on the quiz, there was one for specifically, it was a question about the plasmic reticulum, but the options were either rough or smooth. And in the notes, when we go over whether it is rough or smooth, it's a little bit confusing on which one.
Yeah, I'd have to look at your specific question, but if you send it to me, I can. Yeah, I'll probably have Sasha help me. She's the one that's been helping me just kind of dial in on the chapter quizzes. Okay, so now we got the signal coming down the T-tubule. On either side, we have the sarcoplasmic reticulum. You tell me, what is that sarcoplasmic reticulum going to do? What's it going to do? It's going to store calcium, but when it sees the signal, what's it going to do? Package the calcium. What's it going to do? Not yet. When it sees this signal, what's it going to do? It's going to release calcium. Where's it going to release it? Into the cytosol of the cell. What does that fancy word mean? Is that into the cell or out of the cell? Into the cell. What structure is within the cell that's going to receive that calcium that came out of the sarcoplasmic reticulum? The cytoplasm. It's going to go into the cytoplasm of the cytosol, agreed. But once it's there, it's going to eventually find what protein to bind to. Troponin. Okay, it's going to bind to troponin. Now you tell me the rest of the story. What's going to happen when it binds to troponin? Perfect. I'll repeat that. Calcium binds to troponin. When it binds to troponin, it changes its shape because it has a new molecule that it just found. Okay, so think of like a shadow puppet. Okay, you're standing on a big sidewalk, the sun's on about right there, and you can see your shadow. Right, your outline. You're like, oh, wow, that's pretty good. Okay, then you pick up a giant backpack, and then you pick up another backpack, and you're wearing a backpack on the front, and backpack on the back. And now you look at your shadow. It looks different, right? There's a conformational change of your shadow. So calcium binds to troponin, changes its shape. As it changes its shape, it pulls tropomyosin with it as it's attached. When it pulls tropomyosin with it, it exposes these active binding sites for the myosin head. These active binding sites are on the actin molecule, which is one of 3 proteins that make up the thin filament. True or false? The thin filament is made up exclusively of actin. True or false? False. What 3 proteins make up the thin filament? Troponin, tropomyosin, and actin. So now myosin has the opportunity to see the binding site. And as was just described appropriately, it has to be in a cocked position where you've taken ATP and you've already cleaved or hydrolyzed, that fancy word is called hydrolyze. You hydrolyze ATP, liberating one of the phosphate groups that stays kind of freely associated, but you have adenosine diphosphate. And it moves from this position, which is kind of a forward position, to a cocked position. That's here. In this position, it actually can find the binding site and it binds. And that is what we refer to as the cross bridge. So now you have the cross bridge formed. You still have 2 phosphates that you can hydrolyze to liberate energy, and that's what stimulates the power stroke. So the power stroke down here, you can see these little arrows, the ADP and the freely associated phosphate group fall off. And in order to detach this, you actually need ATP to bind to pull it out of the binding site on actin. And if you don't have ATP available, what do we refer to that state? There's a special term that we call it. Remember? Rigor mortis. Well done. Rigor mortis, or the rigor complex. You with me? Could you put this process in order? If I asked that on the exam, you might want to be able to do that. Okay? Put it in order. Is the order important? Yeah, it's kind of important. That's how muscles contract. That's a huge hint. Like I'd be able to explain this in order. Okay. Excitation, contraction, coupling. We good? Where does the ATP come in? Oh, the ADP. So ATP is adenosine triphosphate. You hydrolyze one phosphate, you make ADP, adenosine diphosphate. If you were able to hydrolyze one more phosphate, you'd make AMP, adenosine monophosphate. But in this process, you really only move between ATP and ADP because you need the energy for the power stroke to take place. You need the 2 phosphates. So excitation contraction coupling, the sliding filament model, really the sliding filaments are wrapped in there because actin and myosin are sliding across each other. But let me just show you. I think it's in. Yeah. This slide. Then this is kind of dovetailing into the architectural labels that you wanted to go over, right? H zone, A band, all that stuff. So again, some of it's here, like the Z disc. You can pretty clearly see. This is a cartoon on the left. This is an electron micrograph on the right. And how is a sarcomere defined? From what to what? Z disc to Z disc. What's another definition of a sarcomere? Another term that would be true. What's that? Functional unit of muscle. Okay, that could be a really, important couple of details to remember, right? On a tricky A and B only type question. So in this state, the thin filament is shown kind of by these yellow thin lines, and the thick filament is shown by this dark or more of a purple blue thicker line. And the M line is what anchors the thick filament, and the Z is what anchors the thin filament. Now, during contraction, true or false, the thick and the thin filaments get shorter and longer. False. What happens during contraction? Just shout it out. They overlap, they slide across each other. The power stroke is what moves them. Remember the video I showed? So here you've got contracting muscle, and you know it's contracting because the Z lines are getting closer together. And there's an optimal point at which they basically bottom out. The thin filaments can overlap a little bit, but the thick filament is completely maxed out. And that's a fully contracted sarcomere. The sliding filament model was designed in the 50s, or described in the 50s, by a couple of different investigators. Huxley and Huxley were their names. And it's been around for about 70 plus years as a model. It's a sliding filament theory, sliding filament model. I didn't quote the names, but I'm going to be in Alan's video history. Yeah.
Are all sarcomeres and all different muscles the same length, or?
Yeah, that's a great question. I actually don't know the answer to that. The question was, in muscles, are sarcomeres all the same lengths? And I'm guessing the answer is no, but I don't really have an answer for you. I'd have to look that up. And I imagine it varies from individual to individual and probably from one muscle to another, but sliding filament model. Nope. Nope. It has actin and myosin, but they're arranged more like a like if you think of like a fish net or a volleyball net or like a basketball net. So they're arranged kind of more in a sort of like a diamond triangular pattern. More fusiform like their shape of the cell. So they don't have that striated appearance. I mean, this is why skeletal and cardiac have the striations, because where the thick filaments are darker, because they are thicker pieces of tissue. And where the thick filament is not, you have a lighter area in appearance. So you get this dark, light, that gives you the striated patterns. Okay, so let's go to the third question, which was, oh, it's right here. Well, you know what, let's go. I felt like there was one that would be a little bit maybe more obvious. Yeah, I think this one's good. Hey, is this what you're looking for? So you see the Z discs here? So that's the sarcomere. We've got our M line, which isn't labeled here, but that's where the thick filaments anchor. The H band is where you have only thick and not thin. The A band is where you have the thick filament. You've got an I band, which is no thick. So the H band is no thin, the I band is no thick. And then, what was the other one? Z, A, H, M is the only one not shown. I mean, this would be pretty simple to draw on a piece of paper. Remember, I had this as homework for you? Draw a sarcomere and then label the parts. I would encourage you to draw a thick filament and a thin filament. You're like, I am not an artist color. I don't care. Just draw it on your test packet so that it makes sense to you. So you can remember that the thick filament is myosin, and the thin filament has 3 proteins that you need to know. Draw it on your test packet. Draw a sarcomere, label it. And it's the first thing you should do, in my opinion, when you get your test packet. Right? If we're down to like, are we down to the A's? Is this all the people getting the A's right here? These are the A's on the exam right here. So draw, I mean, they can watch the video and get the same hints, but draw these things out. And when you're reading the question, refer back to your drawing. There's nothing wrong with that. It'll be helpful. Because many of you are like, I know the information, but then when you ask the question, I get all confused in my head. Well, before you read a question, write down some things that you know. Before I confuse you. I'm amazed that I have that much power over you. Just write down what you know. So when you read the question, you're like, I'm going to refer back to my drawings that I studied. Great question. The elastic filament. The elastic filament, also known as titin protein, is really the 3rd filament. So it would be most accurate to say in the sliding filament model, we have 3 filaments. We just don't give a lot. It's like, it's sort of like the younger sibling that doesn't get any attention, the middle child. How many of you are middle children? Oh, I love you guys. So I have 4 kids, so technically we won't have a middle, but we sort of, one of them kind of has always been the middle, but just personality, just easygoing, just goes with the flow, doesn't rock the boat, just a sweetheart, right? What was I talking about? Middle child. Oh, elastic filament. Okay. I'm tired. Elastic filament. It's that middle child, but it is the 3rd filament, and its purpose during relaxation, when the head detaches and the ATP binds, is it moves the sarcomere back into its resting state. So it operates like an elastic rubber band. So you contract, you detach, right? You bind ATP and you detach, and then the elastic filament moves everything back for the next cycle. Okay, those are the 3 topics that we had. Anything else? Yeah, like a minute left. Okay, come see me. All right, folks. Again, office hours tomorrow, office hours Wednesday, review session tomorrow night. I'll see you on Wednesday. Ask you.